All right, so we're looking at how, in biology, we tend to look at things that are, well, not always, but we do delve into the microscopic world. But in geography, you get given a scale bar and it tells you how many kilometers that scale bar on the page is. So you're going bigger, you see? But in life science, if I give you a scale bar and we say that's only 10 micrometers, we're going smaller. Yes, we do look at things as big as whales, all right? So if you look at the bottom of your page, it says here that you can measure in, let me just get a page that I can try and move this up a little bit. Why is this thing here? I don't want you. Okay. All right, so it's dimensions of biology. So right, and you've got it in front of you. So right at the bottom, 100 meters, that huge sequoia tree, that redwood that people drive through. Did I tell you about that?
Yes, ma'am.
When we were doing secondary thickening, annual rings, meters, we can measure millimeters, you might measure a mouse in centimeters or millimeters. Frogs' eggs in millimeters, okay? Human eggs slightly smaller, all right? Then we start with the tissue. Those notes that were giving me a problem just now, the machine was doing crazy things. And we start looking at micrometers. Then we start looking at nanometers when we're talking about our dear old, not so much friend, COVID. Okay, nanometers, and then eventually there's even things like picometers and so on. And you can start looking, working out the size of atoms and amino acids and proteins. And that's what we've done this year. We've worked our way from carbon, hydrogen, oxygen to amino acids, making proteins and starch and glucose and all of those. We then looked at cells, we've looked at some tissues, and we're eventually going to look at, next week, animal tissues and, the skeleton, the muscles and the movement. Have you managed to get hold of him?
Yeah.
It's just me here.
You gave me a missed call.
Okay, all right. Okay, so that's what that page is about, but in the little...
Max says you must come on.
Kian! Kom jong.
Jan says there's no one on.
Just tell him I did the Google Meet late.
Yeah.
Okay.
I'm saying
You're awake.
yeah, it sounds like he just woke up.
I had to phone someone last term for some life science thing and I woke them up.
Imagine your teacher.
And I knew I'd woken them up because they were late.
Oh, no.
And they're like,
But
yes.
you don't know, just leave a number there, sure, because they wouldn't have your
No, I phoned from the school phone.
Oh,
You
no.
emailed.
I had to phone him with emergency. Okay. All right, guys. So, just going back now, we're talking about the system international, that wherever you go, if you're a biologist and you go everywhere, America uses pounds and all that stuff, but when they, scientifically they would use micrometers and millimeters. I still don't understand why they're still on pounds and ounces and all that kind of stuff. I mean, that's what my mom and dad grew up with. You know that, hey, in South Africa.
Pounds.
And then when we were born, then they went over to the system, or the metric system, with meters and millimeters and the standard system. Didn't you know that? But here, it was pounds and ounces when my granny and my mom and them, that's what, and they used to, the money wasn't rands and cents, it was tickies and dimes, from England. It was that. All right. That was before I was born. Okay, so 1 meter is how many centimeters? You can make notes on your sheet, whatever you want to do. How many centimeters? I'm just getting into it. How many centimeters? How many centimeters in a meter?
100.
Thank you. All right, but if now, how many millimeters in a ruler? In a meter? 1,000. So we're working from a millimeter down. We're going deep. We're going down now into the microscopic world. All right, 1 millimeter on your ruler. Here, 1 little millimeter. Do you know that? Well, you know it because I told you before lockdown. Is divided into 1,000 micrometers. And sometimes they're called mu. Okay, so it's little u, it's mu. It's a Greek letter in mu, M-U. It's got a little tail and then like a cursive U. And sometimes, the metric system we use micrometers, but sometimes you might see microns. If you see that, then it's the same. All right? Then, so if you take 1 micrometer, Okay, so if you divide the millimeter here into by 1,000 and then by 1,000 again, so in other words, by 1,000,000, you then get nanometers. Alright, so that's what we're doing here. So on this picture, you can, human egg cells and frog egg cells, you can see they're millimeters. You can see them with the naked eye. But when you start looking at epithelial cells, red blood cells, parenchyma cells, all of those, you're going to start using micrometers. There's 1,000 micrometers in a millimeter, but when you start looking at our lovely friend COVID, you're going to start looking at nanometers, which is millionths of a millimeter, thousandths of a millimeter or millionths of a meter. Making sense? Okay, so the part that we need to know is this. Alright, what is a micrometer, what is a nanometer, and when you measure with your ruler, what units are you going to use? Never, you must never use centimeters, because then your conversions are going to be crazy. Because I mean, here, look, we jump by multiplying by 10. So please don't measure in centimeters, because then you start making silly mistakes, and it's not going to work so well. All right, so I'm still lonely, Kian. Where are you?
You said you were material on that.
Hey?
You said material and you were on
I'm not seeing anyone here.
Did you join back in the same meters then now, or did when your friend said
Did you join?
Well, it only gave me one meeting active, and it said rejoin. Okay, let me just check the pin. Over here, I need to go into calendar. Oh, there it is. So, okay, no, okay, wait, I'm making you one. Why didn't you make it? Because I've got it on my phone. That's really odd. Okay, so it's now 8:40. Let's do a new one. Okay, L is 11 September, choice 3. And now 8:40 is not this one, so we'll change it to 8:42. We'll leave that. We'll add guests. 10. Choice 3. Over there, 8:42. Save.
Mama, you're sending it to the right choice.
Choice 3. No, I think that is not. For some reason, it just didn't want to. Okay. Now, just look for my. I'm just looking for a sheet that I was doing now. It's gone. It's a PDF. It's opening. Okay. If they missed that part, I taped it yesterday as well. Okay. All right, I'm joining the new one. You can tell them I'm joining the new one. I am the first here. Okay. All right. Can I just say that again? No. Okay, so guys at home, you can go and watch the beginning of choice 1 video, because it seemed like I wasn't, I don't know, the wrong Google Meet. Made a new one now. So you can watch the beginning of that, and now I'm going to start looking at the calculations on magnification and scaling. Okay, so are we happy with that so far, about how we get to all of those and which ones you need to know? Now at the back, do you remember doing this? So it's the whale worksheet, all right? We just explained why we need smaller and smaller units of measurement. Then at the back of that, I gave you the formulae for either if you are given a scale bar or if you're told how many times magnified the photograph is. Okay, so whether you are given, say, times 130 or you are given a little scale bar with something written above it. Just like in geography. Okay, so it's this page now. There, with the triangle. Alright, so do you want me to go through all those? Calculate. Well, I think I should. Alright, so let's go through them again. And we'll work out how the formulae work and how we get to it. And we'll do all of that again. Here's my answers. Okay, so you see there you've got a triangle. Alright? And the triangle is, have you ever used that in science? Like, for example, if you do density equals mass over volume, you can, and then you can work out all the other, have you ever used that?
We've done it in math.
Yeah. And the other class said, no man, they tell us not to use that because whatever. I don't know why. Okay, so what I've done here is I've used, okay, so on the sheet you've got the formulae here, then some questions, and then what I've explained on the board just now about millimeters and micrometers, there's a summary, okay, but what I've given you is fine. Then at the bottom you've got a picture of a parenchyma cell, a red blood cell, a rod-shaped bacterium, and a spiral-shaped bacterium. So I was just using that as practice, okay? So, if we look at, it says use on page 2 now, it says, please just ask them to get on again. Yeah, because I'm now on the new one and I've just sent another email and I don't want them to miss.
So we should get the email then.
Everybody should have got it, yeah. And they, yeah, okay. It's weird that they couldn't get onto that first one.
I made a new one.
Tell Mateo.
Yeah, tell Mateo.
Who else? And Tizzy. Good. Who can tell Tizzy?
Just send it on the group.
Do you have a group?
I don't have a group, man.
Hey? Well, then put it on there, please. Say they've got an email. Hello, Mateo. Sorry for the mix-up. Alright, but you can watch the beginning of the Choice 1 video. And this part I actually didn't do. What we're doing now, I didn't redo in Choice 1. Are you happy that I go over it again? Alright, so go and find your whale worksheet, scaling and magnification. Alright? The first part I've explained already, but it's taped on Choice 1 from yesterday. And now we're going to do page 2 with the formulae of how to do scaling and magnification using the diagrams on page 3. Okay. Alright, so it says there, use the diagrams on page 3 to calculate. Okay, so number 1, it says if the parenchyma cell is magnified 130 times. Okay, if it's magnified 130 times, what is its actual length? Now, you might be asked to do, okay, here's the mitochondrial model. So you might be asked to do actual length, actual width or diameter, or actual something. So if I've got a very big model, and I know how many times it's been magnified, I can work out how small it is in real life, yes? And that's actual length, actual size, actual whatever. Okay? So the formula for magnification. Now, Mrs. Page was going red in the face with the grade 11s. And we were going red in the face with the grade 12s. When you write down a formula, you've always got to write down the formula first in your tests, whatever, pracs, and then you can use abbreviations. But you've got to write it down first in full. You can't use, because I do use abbreviations eventually, but people forget that I say, the first time you write it out in full. Now, magnification if I have got this mitochondrion and I know its actual size, what's the formula for magnification? Working out how big it is, how many times it's been magnified.
Actual size times measured over actual.
Okay, yes. All right, how big it is on the model or on the page, I know it's there, but I want you to also think about it logically. So it's measured size over actual size. Right? So in other words, this is in the photo and that is in real life. And in the photo or my model is huge, but in real life it's going to be really, really small. And if I divide by real life size, I'm going to get to the magnification. Do you send out a message? Okay. To Tizzy as well. Anyone?
I'll send a message.
Have you got her phone number?
Which is she on the group? Yeah.
Please. Mateo, is anyone else there? Mateo, is anyone else there? I can only see your icon. Yeah, I know they're missing because there was a mix-up with the Google Meet. Okay. All right, but now the question has said to me, if the parenchyma cell is magnified 130 times, what is its actual length? If that is magnified 130 times, what is its actual length? What do I do now?
Actual size.
So you swap. Okay, guys, please just sound a little bit alive. So it's actual size or actual length or whatever it is. The formula is now going to be.
She's checking it's the right link.
Okay.
I put it on the group.
Look for the new email.
Yeah.
Okay, so actual length is equal to what? Measured size over magnification. But that should make logical sense because I've got this big thing, and if I divide by how many times it's magnified, I'm going to find what its real length is, how small it is actually in real life. Guys, what did you get there? What was the length of this parenchyma cell? Just have a look again. I know I've got it here. Yeah?
77.
Yeah. If you measure from there to there, you got 77 millimeters. Whoops. And if you divide by 130, our answer was?
0.59.
Or 0.6. Okay, 0.59 millimeters. You can abbreviate that to 0.6. So what we're really saying is, the parenchyma cell. This parenchyma cell in real life is 0.5 millimeter long. Yes? Doesn't that make sense? Think of how small they were when you look under the microscope. Okay, so that's question number 1. Now we're going to use the scale bar. Okay, so the scale bar given here at the top of this picture on page number 3, it says that length is equivalent to 10 micrometers in real life, just like when you're using a map and it tells you the length of a bar is equal to 40 kilometers and you're trying to get to Durban or wherever it is. Okay, so question number 2. Using the scale bar provided, calculate the actual width. So now we're looking at width of the parenchyma cell. Okay, do you remember me showing you how we get to this weird formula? Okay, so if I look at this parenchyma cell, here it is, okay, there's the nucleus, there's the cytoplasm. We are trying to determine its actual width. Okay, so if I look at that, do I know the actual width? No, but I know the measurement on the piece of paper. So, okay, I'm going to show you how we get to that formula. Okay, there's my little column headings. Okay, what is the width from there to there? Measure it in millimeters or read your answer from before. 58 what? Millimeters. Okay, so from there to there is 58 millimeters. Alright, do we know, hang on, what am I doing here? Yeah, do we know its real life width in micrometers? No, that's our x, that's our solve for x. The scale bar, do I know how many micrometers it represents? How much is it? 10. Do I know the length of the scale bar?
Yes.
Measure it. Okay, Shanae, so you measure the length of that scale bar. And how much did you measure it as? 14 what? Because you always measure in millimeters. So all I'm doing is using logic. I'm saying, I know that that parenchyma cell across is 58 millimeters, but I don't know its real life width. But I've been given a scale bar, and I know that that length represents 10 micrometers. So I put it in the micrometer column, and I've measured it, and it's 14. So all we're doing is solving for x. So if I want to solve for x, how do I move the micrometers, the 10 micrometers across?
Multiply.
You multiply, so you're going to move it to this side, just like maths, okay? So times 10 micrometers, you don't have to know how to do this, and divide by 14. Okay, so now where do we get the formula from? What was the 58? It was the measured width, or the measured size. What was the 10? It's the number on the scale bar. What's the 14? It's the length of the scale bar. Okay, it's the length. Let's write it out in full the first time. It's the length of the scale bar. That's what my formula is. If I'm using a scale bar to work out actual work length or whatever, you say measured size times whatever's written above the scale bar divided by the length. So you've got a length at the bottom and you've got a length that you've measured.